So let's start this session that addresses a broad panel of problems related to sequencing data analysis. Our first speaker is Vincent Sater from Rouen, who will present us UMI-Gen, a UMI-based read simulator. Vincent, to you.
Thank you, Raluca. Hello, everyone. Today, I'm going to present to you a new tool called UMI-Gen, the first UMI-based read simulator that we developed at the Litis lab in Rouen, and that allows better benchmarking for both raw read-based and UMI-based variant caller. I'm going to start with a little reminder about the big steps in an NGS experiment. Normally, we start by extracting the DNA, fragmenting it, and amplifying it via PCR in order to obtain enough fragments for the sequencing. The amplified DNA fragments are then sequenced by an NGS sequencer, giving us the results in a FASTQ format, which is a file containing the sequences of the DNA fragments. So what are UMI? UMI stand for Unique Molecular Identifier. They are short arbitrary nucleotide sequences that has been increasingly used in next generation sequencing since 2015. So now we need to introduce these UMI tags in our DNA. For that, we start the same way by extracting and fragmenting the DNA. However, this time, before amplifying the fragments, we are going to attach one unique UMI tag to each fragment in a known position. For example, at the start of the fragment. After introducing the UMI tags, the steps remain the same, but a new step is needed in the end in order to extract the UMI sequence from the sequence of the fragment, allowing us to use them for the downstream analysis. For the sequences to be used efficiently, they have to be aligned to a reference genome, which can allow us to detect variants in our DNA fragments. A variant is detected when the sequence based at a certain position is not that of the reference genome. So theoretically, if a variant, here shown in red, if a variant is present on the initial DNA, it will be tagged by the UMI attached to the fragment. Every time the fragment is amplified, the variant and the UMI are amplified as well. Therefore, on each fragment tagged with that UMI, the same red variant should be seen. On the other hand, during the amplification and the sequencing steps, PCR and sequencing errors occur in the fragments. In this case, the error is specific to the fragment on which it occurred and not to all fragments with the same UMI group. This is how using UMI tags allow us to distinguish between true variants caused by a mutation on the DNA and false positives caused by PCR or sequencing errors. If the variant is found on all the reads tagged with the same UMI, then it is a true variant. However, if only some of the reads tagged with the same UMI group present a variant, it is considered as an artifact. When we want to compare the performance of a variant caller with that of other tools, we are often faced with the problem that the data used for benchmarking is unknown to us. This means that we don't know the number of real mutations in the used samples. So we can't calculate the number of false positives, false negatives, true sensitivity, nor true specificity. This eventually leads to a biased and unfair comparison. The solution, of course, is to use a read simulator to produce artificial samples that mimic real tumoral samples, and in which the number of mutations is known and controlled. At the time, a UMI-based read simulator didn't exist, so we developed a UMI-Gen. So UMI-Gen uses a genome reference file in the FASTA format and a bed file containing the coordinates of the targeted regions in order to create UMI-tagged reference reads. These reads present the reference base at each position. Afterwards, using a set of control samples, UMI-Gen estimates the background noise of the sequencer and inserts it into the referent reads. Finally, if the user wishes to add variants to the simulated data, he can do so by providing a CSV file containing a list of the variants with their precise positions and allelic frequencies. In order to estimate the background noise, UMI-Gen starts by generating a pileup on the control sample. The pileup simply contains the counts of A, C, G, and T at each position on each chromosome. A pileup is generated based on each control sample, and they are used to produce an average pileup based on all the samples. Then, since variants can still be found in the control samples, UMI-Gen includes a variant calling step to remove potential variants from the pileup. The third step consists of calculating the probability of seeing each of the bases at each targeted position. This is easily done by dividing the number of occurrences of every base by the depth of the position. Finally, UMI-Gen calculates an average quality score of the position by analyzing all the reads covering a certain position in every control sample. Now we reach the step of inserting the variations. In fact, inserting an artifact or a true mutation requires modifying the frequency of the alternative base to a specific value. Therefore, in order to insert a variant as an artifact, the tool will seek the position in question and will change the reference base to the alternative base on a number of reads, allowing for the desired allelic frequency to be reached. The same strategy is used for inserting a variant as a true mutation, only this time the UMI of each modified read will be changed as well. All the modified reads will finally be tagged with the new variant-specific UMI. We use the same in order to validate our pipeline, we used UMI-Gen to generate two simulated data files, sample 1 and sample 2. Sample 1 has a depth of 1000x and contains 13 true variants with frequencies from 1 to 90%. On the other hand, sample 2 had a depth of 10,000x and contains the same 13 variants from sample 1 plus 2 variants with frequencies of 0.5 and 0.1%. So we used the same 6 control samples to reproduce the sequencer background noise in both samples. This table presents the pileup of a certain position at chromosome 1 across all samples. Using counts and the depth of each sample, UMI-Gen can then calculate the probability of having an A, C, G, or T on every sample. Then, using these numbers, it will calculate the average probability of each base at that position. These average probabilities are used to reproduce the background noise at the specified position in the final sample. For example, sample 1 has a depth of 1000x. Multiplying the probability of each base by the depth of the sample gives us the number of reads that should be presenting every base. So for sample 1, the noise at this position will correspond to 1 for base A, 4 reads showing a C, 6 reads showing a G. On sample 2, the depth is 10,000x, which leaves us with 8 reads for A, 44 reads for C, and 63 reads for G. Once the files are generated, we use the tool IGV to check if the noise is present in the final reads, and we found it in both samples at the expected position and frequency. We also used IGV to check if the variants inserted as true mutations were correctly inserted in the files, and we found all the variants at the correct frequencies on both samples. UMI-Gen can also reproduce the base quality score per position in reads in the simulated data. We compared the variation of the quality scores in the control and the simulated samples and found it to be very similar. We then simulated a loss in base quality in the control samples at position 85 of the reads, and as expected, UMI-Gen reproduced the same quality drop in the simulated data. Finally, in order to verify that the produced samples mimic the true control samples, we compared the GC percentage of the two datasets and found it to be nearly identical on both. Once we verified that UMI-Gen can generate simulated data that resembles real-life sequence data, we compared the performance of 4 variant callers using sample 1 and 2. We use SymVict and Outlier as raw read-based variant callers that specialize in the detection of low-frequency variants. We also use 2 UMI-based variant callers, Deep SNV Miner and our own UMI Varcol. SymVict and Outlier had a fairly good performance on both samples. SymVict was able to detect 8 of the 13 and 8 of the 15 variants in sample 1 and 2 respectively, while Outlier managed to detect 11 of the 13 and 12 of the 15 variants on the same sample. However, the number of false positives was really high for both tools, especially when the depth increased. On the other hand, Deep SNV Miner was capable of detecting 12 of the 13 variants in sample 1 and 14 of the 15 variants in sample 2. In addition, the number of false positives was relatively low, and it actually decreased when the sample depth increased. On sample 2, for example, Deep SNV Miner only detected 1 false positive. Finally, UMI Varcol managed to detect all the variants on both samples with 0 false positives for both configurations. These results allow not only to validate the data generated by UMI-Gen, but also to demonstrate how using UMI tags in variant calling applications can be very crucial for the detection of somatic variants at very low frequencies. Concerning the performance of UMI-Gen, we tested the tool on a number of simulated data with increasing depth. 2 minutes with 1 gigabyte of RAM are needed to generate a sample with a depth of 1000x, while 18 minutes and 5 gigabytes of RAM were required for depth as high as 10,000x. In conclusion, UMI-Gen is the first UMI-based read simulator capable of producing simulated samples in which real variants are known. Most of the parameters are customizable, which allows total control over the simulation. UMI-Gen generates the simulated samples in FASTQ, BAM, and SAM formats for each sample and is available on GitLab for Python 3 use. Thank you for your attention.
Thanks a lot, Vincent, for your nice talk, and we'll have the time to take some questions. So the first question is from Pierre. Hi, Pierre. In fact, I had the same question as he does. So how do you take into account, sequencing errors in the tags themselves? And to add something to these questions, I'm curious, do the variant calling tools work with this kind of errors, take into account this kind of substitutions?
For the first part, no, UMI-Gen does not take into account errors in the UMI sequences. There are tools that can correct errors in the UMI sequences, but in fact, when we try to estimate the error rate in the UMI sequences, we found it to be relatively low. So we didn't bother adding this to the algorithm. For the second part, can you?
I guess you just answered. It was just to complete it. Thank you. For the moment, there aren't any more questions in the panel, so I'll ask a second question. I was thinking about RNA-seq, and so using UMI tags in RNA-seq. Did you think about simulating this kind of reads in this context?
In fact, we developed UMI-Gen to test our variant caller UMI varcall. So we wanted to use a simulator that existed instead of developing one ourselves, but there weren't any, so we decided to develop a UMI-Gen. However, for the RNA for RNA-seq experiments, there are already developed tools that allows to simulate data sets with the UMI tag.
Okay, thank you for your answer. So I don't see any more questions in the panel. I say to thank Vincent for his talk and his answers to the questions, and we'll skip to the next speaker. Thank you, Vincent.
Thank you.